welcome you, especially our neighbours, to the Dorcus Cummings lecture. Many of you are here for the first time as guests of the people who know the laboratory. So this, we have a kind of an interesting audience here. We have a mixture of people from the local community, but also some of the world's greatest cancer researchers. And because we're in the middle of a symposium, actually towards the end of the symposium, on cancer therapy. This is part of the series of meetings that Cold Spring Harbor Laboratory has each year. We have about 30 meetings in this auditorium and meetings elsewhere on the campus and another campus in Lloyd Harbor. And this meeting, the Cold Spring Harbor Symposium, is the premier meeting of the year, and it has been going since 1933. And the tradition has been that the scientists who visit here have been invited to the homes of the local community. That tradition started way back in the 1920s, in fact, before the meeting started, when scientists came to Cold Spring Harbor to work over the summer, when they didn't have to teach in universities. They did research here and were invited to the local neighbors, including one famous event in 1932 where scientists were invited to the Marshall Field estate, a 3,000-acre estate just up the road that was owned by Marshall Field at the time. And he had a dinner party, and there are photographs from that, and it was attended by Fred Astaire and Ginger Rogers. So hopefully maybe you'll see somebody of that fame tonight. So I want to thank the hosts and hostesses for the dinner parties tonight for hosting these events, and it has also been a tradition to have a speaker chosen from the scientists and clinicians who are speaking at the meeting to give the kind of public lecture, so called the Dorcus Cummings lecture, and we're very pleased that Robert Cummings and his wife Poppy are here, and his sister is going to come soon, I understand. So it's great that they could be here as well in honor of the lecturers and in honor of their mother. Tonight's speaker is Charles Sawyers. He's at Memorial Sloan-Kettering Cancer Center in New York City, and he's also an investigator at the Howard Hughes Medical Institute. But he is one of the most well-known cancer scientists in the United States because of his combination of, he is a clinician, combination of clinical work, but also his really pioneering science discoveries. He went to Princeton and obtained his medical training at Johns Hopkins University, and then for about 18 years, I think, was at UCLA before being recruited to Memorial Sloan-Kettering. And he's had a really fantastic career and has had a major impact in cancer research and also therapy. He was a part of the team that developed the really now famous drug Gleevec, which has been a transformative drug. He may mention that. In fact, I don't think Charles can give a talk without mentioning that. For that work, which he did along with a couple of others, he won the very prestigious Lasker DeBakey Award, which is the most prestigious medical research clinical prize in the United States. He has also won a number of other awards, including the rather lucrative Breakthrough Prize, which is a $3 million prize for scientists. And, but more importantly, he's had a very, very big impact on therapy, and most recently he's been focusing not on leukemia, which is the drug that Gleevec targets, but on prostate cancer, and has developed new therapeutics for prostate cancer. In addition to this great science and clinical work that Charles does, he's also been very, very prominent in the kind of society aspects of science. He chaired a very prestigious committee of the National Research Council, or the now Academy of Medicine, that looked into how we should move healthcare and research forward, and was the chairman of a committee that wrote a very influential report on what is now called precision medicine, and he'll explain that. More recently, he was tapped by Vice President Biden to be on a member of the Moonshot Cancer Committee that got together a committee of clinicians and scientists to advise the White House, and particularly Joe Biden, who has a very, very strong interest in cancer now, advise him on how we should rapidly move forward with new resources in approaching and treating cancer. And Charles has also had a major role in Stand Up to Cancer, which is now a large fundraising organization for supporting innovative cancer treatment research, and also the American Association of Cancer Research. So it is really a privilege to have Charles here to give a talk, the Dorcus Cummings talk. It's on precision medicine and moonshots and other things.
Thank you, Bruce, for that kind introduction, and thanks to all of you for giving up your late Saturday afternoon. Show of hands, I'm going to assume that most everyone in the audience went to high school, and because I'm going to show some slides. Some of the slides that I show were actually part of a talk that the Howard Hughes Medical Institute asked me to do. They spend a lot of effort trying to disseminate science into the high schools in the Washington, D.C. area. The headquarters of the Howard Hughes Medical Institute is in Chevy Chase. So I want to give them a shout out for this. That's called the Holiday Lectures on Science. So you're going to hear a talk that's geared to you with a high school biology background. And I'm going to stop and ask you questions in the middle. So no falling asleep. The timing is actually quite good. So the term precision medicine was actually first coined in this report that Bruce mentioned, which we published in 2011. Of course, a lot of these reports sit on the shelf and gather dust, and this one was doing that until January of last year, when President Obama declared the Precision Medicine Initiative at the White House. So it took a little time to get the wheels of government to buy into this. And I'm sure some of you have read about it, and I'm happy to answer questions. Now, the cancer moonshot thing is a more recent development. Many of you, I assume, watched at least part of Obama's State of the Union this past January, when in the middle of it, with no prior announcement, he turned around and looked back at Vice President Biden and said, you know, it's time to cure cancer, and I'm putting you in charge of mission control. So for the last 4 or 5 months, the Vice President has been busy living up to that job, trying to get advice on what to do. And as Bruce said, I'm one of 36 people on a panel where we are on conference calls frequently, and you know what it's like to be on a conference call with 36 people. So I'm happy to tell you what we're talking about in the end. So let's start with first principles, but maybe even before that, I want to leave you with 2 or 3 main things. The first one is, this is an incredibly exciting time for cancer research, and you will see why. The second is, this has been a long time in coming. It required a fundamental, years and years of commitment to understanding basic cell biology and cancer biology, largely due to work at a place like this. And the third is, the moonshot is there's not a single moonshot. This is a problem we're going to have to live with for a long time coming, and we have to sort of avoid the hype of most new media articles on a new discovery saying, oh, aren't we almost there? Because I don't want to, take the wind out of the sails. It's an incredibly exciting time, but we have to manage expectations. So let's start. So cancer, you all know the term benign and malignant. So benign means a tumor in green is a proliferation, starts in one cell, it starts to proliferate inappropriately, but it just sits there. It's not causing a big problem, unless it gets really big, and then you just ask a surgeon to take it out. Starts to become a problem when the initial tumor cell locus cells acquire the ability to escape sort of the mothership and start invading into tissues. And then the real problem, of course, is if it gets into the blood, then it can metastasize to other sites. So the top one is not cancer. The bottom two are cancer. How do we treat these cancers? You know many of these things. We rely on surgeons. Surgeons cure cancer. They are obviously, that is the best way, and we have to detect tumors early enough in order to have that work. The two other main therapies you've known about and you've had friends who've had these therapies are radiation and chemotherapy. These kill cancer cells by damaging the DNA in the cells. They kill normal cells too. Neither a normal cell and a tumor cell both require DNA to replicate, and these two therapies work by doing that. The problems of, the reasons that we need more therapies is a surgeon can't see cells that have escaped the tumor, and neither can a radiologist, so they can leave cells behind. And as I said, these two therapies don't discriminate well enough between normal and tumor, and therefore there are a lot of side effects. So the excitement over the last now 15 plus years is from this, and this is the DNA sequence. So let me show you how that's happened with some examples. So the main point is that we now know that cancers are caused by mutations in our normal genes. We call these cancer-causing genes. We've now adopted the terminology of drivers. These are mutations in a gene that now become the driving force of the cancer. And there are two flavors. There are those that we call oncogenes, which we use the word dominant. This is Mendel and the peas, dominant versus recessive traits. The other category are tumor suppressor genes. So this is a cartoon that depicts the cell cycle. Cells, when they divide, go around this cycle. An oncogene is a go signal. A tumor suppressor gene is a stop signal. If you get a mutation in one gene that's an oncogene, it sends a go signal and the cell divides and you form that little tumor. For a tumor suppressor gene, one mutation doesn't do it. You have to get rid of one as well as the other. Now, some of you may know that these tumor suppressor genes are sometimes inherited. So the famous example is the breast cancer gene BRCA1 and BRCA2. And families who have that mutation in their germline, meaning in every cell in their body, and can pass it on to their children, have one mutation already. So the reason those patients people are at higher risk is they only have to have one mutation because the other one's already gone in order to get the tumor. All right, so a very simple concept is if you have a mutant oncogene and you know that that's driving the cancer, why not make a drug that inhibits that mutant protein? So that is what's called targeted therapy. And the example that Bruce mentioned is this drug called Gleevec. So here's the drug. It's a chemical. This is the structure. And here is the blood smear of a patient who has chronic myeloid leukemia. There are too many of these white blood cells in this patient. And why are there too many? Here's a normal, and here's CML. Well, if you use a technology called cytogenetics, where you grow these cells in the laboratory and look at the chromosomes, these are lined up. You have 1 to 22, and then here's the X and Y. This is a male patient. If you're really good at looking at these all the time, you can see that this 22 is too short. This discovery was made in 1960 in Philadelphia, and this is called the Philadelphia chromosome. That's a pretty subtle change. But this is the power of observation. It took another 10 to 12 years of developing technology to recognize that what was actually happening is that the missing little piece here is actually now over here. And what's shown, what basically happened in one cell in the blood is that there was a break at the bottom of chromosome 9, there was a break at the bottom of chromosome 22, and these two pieces switched places. So what? Well, turns out that break is in the middle of a gene on chromosome 9 as well as on chromosome 22. As a result, the gene on chromosome 22, which goes by the name BCR, and the one on 9, which goes by the name ABL or ABL, forms this hybrid gene which creates a protein which is half BCR and half ABL that does not normally exist. And what I'm showing you here is actually what that protein looks like at atomic scale. We have solved the crystal structure of this protein. We know that this is true. I'm going to show you a couple of movies of what this looks like. So what is this? This protein is a machine. We call it an enzyme that is executing a function. It moves. You can see that here. This is exactly what it looks like, but with a little bit of animation. If you take if you look sort of at the skeleton of the protein, you see this loop that's moving back and forth between what's known as an active and inactive conformation. So now let's look at it slightly differently. So now the protein is floating in the guts of the cell, the cytoplasm, where it's now doing its thing. It's moving back and forth. What does it actually do? Well, it binds a molecule called ATP that's in yellow. It then transfers a phosphate. ATP is adenosine triphosphate. It has 3. It transfers 1 to this other protein called a substrate. That's a signal. Think of this as a chain reaction, kind of a bucket brigade passing a signal from one protein to another. It turns out this process of using ATP is a commonly used process by many different proteins. So that's what it does. What can we do about it? Well, now that we know exactly how this works, can we make a drug that would, gum up the works of that process, but do it very precisely and not do it to every protein that uses ATP? Because ATP is used by lots of proteins, over 500. So what happened in Basel, Switzerland in the early 90s were some chemists decided to look for such an inhibitor. So you could imagine, what I've shown you, we actually didn't know in the early 1990s. A lot of that happened later. You could imagine if you knew what I just showed you, maybe you could just draw on the board exactly what chemical would fit in that pocket where the ATP was binding. It turns out, even with what we know now, that's still incredibly challenging. So the approach that was used more than 20 years ago is still the most efficient way to at least get the first look at what the chemical will look like. So here is a library of chemical structures that chemists have made over years. So what happened was, let's now search through this whole library, millions of them, to see if we can find one that will block BCR-ABL. And that's exactly what happened. It can be done through robotic technologies. It happens in the matter of months that you can sift through large chemical libraries. And then Gleevec popped out. And now, let's go back to the movie and see what happens. So here's the protein again. Gleevec is now in purple. It slips right into this position where the ATP, the yellow molecule, would have been. And what's really clever about it is it slips in and locks this activation loop in what's known as the closed conformation. So it tries to move over, and it can't. So it's completely disabled the signaling. So what happened when this drug went into patients? Well, I think you know a little bit of that story. This is a This is the data from the very first clinical trial that I participated in. 6 patients who got the drug, and the blood count, which is shown here in the 50 to 100 range. Normal is in this range here. All the blood counts came down. Patients with this disease who were first treated on clinical trials in the 1999-2000 timeframe, some of those patients are still alive and well, taking this pill once a day and living a normal life. All right, so that is sort of that is what targeted therapy is. That is what precision medicine is, because this pill works in patients who have the BCR-ABL mutation, but it doesn't work in patients who don't have the BCR-ABL mutation. So, are you with me?
Thank you.
All right, because I'm going to ramp it up now. Because this really led to a series of conversations amongst the whole field. In fact, I remember being in this auditorium probably 15 years ago saying, should we have a moonshot? We didn't use the word moonshot, but is it time to do something really big? Is there sort of an engineering approach to push this? How many more examples of this are there? I mean, just to reflect. It started in 1960 when someone was looking through a microscope, Peter Knoll, and saw that pattern. It was 40 years later when Gleevec was approved. We can go faster. And the thing that enabled us to think about going at large scale was the ability to sequence DNA at great speed and low cost. So a little primer on DNA sequencing. So DNA sequence is a string of 4 letters. A mutation is a misspelling. The spell check function didn't work. We can do this through these new technologies, which is called next generation sequencing. This is a little, it's now 1,000. This was 2 years ago. The decline in sequencing costs are way faster than Moore's law, which is the doubling of transistor size and half the cost from silicon chips. And so what's the big idea here? The big idea is that let's, instead of relying on cytogeneticists to look in a crude way at chromosomes, let's sequence the DNA base by base, all 3 billion base pairs in a tumor, and compare it to the patient's normal DNA and see if we can find mutations. And let's do this for thousands of tumors and see where we stand. So I'll also remind you that the reason these mutations create a problem is because the DNA sequence, the so-called triplet codon, leads to an amino acid. And there's three bases here. If the last one changes, that's called a silent mutation because a tyrosine, this triplet codes for a tyrosine, that's an amino acid. This mutation is what's called silent. So if we counted all the silent mutations from this giant sequencing project, we might get confused. Maybe those mutations shouldn't matter. But the two that we're most interested in, at least then, were missense mutations. So just A to a G changes this tyrosine to a different amino acid. That can have a very profound effect on a protein. And the other is there's a codon change. If this C turns to a G, it becomes a stop codon. The translation of protein from the messenger RNA that this derived from just completely stops. The protein doesn't get made properly. That's a very profound disruptive change. Okay, so what ended up happening is through a national effort run by the National Cancer Institute, as well as an international effort, the community of DNA sequencing community and cancer biologists got together, and we've sequenced well over 10,000 tumors. What did we learn? Well, we can generate graphs like this, where we can count the number of mutations we found in a tumor. and we can now start to look at some patterns. So there's something really interesting here that just should grab you right off the bat, and that is children with cancer, their tumors have very few mutations. Adults have more, and there's a wide range from 25, 100 to thousands. Does this matter? And what's underlying this pattern? So here's the adult ones. Let's look at the names of some of these cancers. This is a really interesting result. So we think of lung cancer as lung cancer. And we know that smoking is associated with lung cancer. If you get lung cancer and you weren't a smoker, you have very few mutations. If you have lung cancer and you are a smoker, you have 100, 150, 200 mutations. This makes perfect sense. We know that carcinogens in tobacco smoke cause DNA mutations. So we're not learning anything that we didn't suspect, but now we have real quantification of this, and as you'll see, it's going to matter a lot. This one's an interesting one. This is melanoma, skin cancer. Again, once we saw this, it made perfect sense. Skin cancer is caused by exposure to sunlight. UV light causes DNA mutations. That's why we wear sunscreen. This one's actually quite interesting, the one that's in the thousands. It took a little while to sort this out, but this is primarily a subset of patients with colon cancer who have an underlying mutation in genes that are part of the spell check. So if you disrupt the spell check, you're going to, the error rate is going to go way up, and that's exactly what happens here. And it turns out this makes a big difference for therapy as well. All right, what I've just described to you is the mutations. How do we know if those mutations are in genes that are important in causing cancer? Are they drivers? So how do we sort that out? We still don't know the best way to do this, but today, in a very good way, is to count them and where they are and in what genes they are. So think this is a grid that lays out each chromosome from 1 to 22 and then the X chromosome. And imagine this is the beginning of chromosome 1 and this is the end of chromosome 1. And let's start saying, where are these mutations? And you start seeing that there are these like skyscrapers of mutations, these towers or mountains. And these are genes that we know, and in many cases already knew, were cancer genes. And then the ones that are smaller in size are clearly cancer genes because we keep seeing them again and again, but they are less common. And in this way, a statistical strategy, we can determine which ones are most likely to be the true drivers. So now that we've done so many thousands, where do we stand? Well, turns out there's quite a few. So here are the names of a lot of these, and the size of the print is how common they are. The most commonly mutated gene is a gene called TP53, and some others as well in here that some of you may have heard of, and we have been hearing about the nitty-gritty details of how these proteins work for the last 4 days at this meeting. And it's very cool, exciting, and as you'll see, promising for therapy. So do we have them all? Well, we can't really know probably ever if we have them all, but we certainly know we have the common ones, and common is defined statistically as present in at least 5% of the tumors of that type. So if we sequenced another 100,000 tumors, there's no question we will find more. But they're going to be quite rare across the population of cancer patients. So I think the good news about this is that we know our enemy in way better detail than we did 10 years ago. And yes, it's challenging, it's several hundred, but it's not thousands. And as you'll see, we can bucket these 100 into small groups that hopefully common therapies can be applied. But there's no doubt that cancer diagnosis will include and is including sequencing to find out what's the status of these genes in your tumor, because it really matters. So here's a way of bucketing those hundreds of genes into a few categories. So I'm going to tell you about two of them. One is just simple to understand. It's the go signal that I mentioned before, cell growth and survival. The other, a pretty big category that we sort of knew about, but now that you see the whole complete map of this, these are genes that are normally involved in what's called cell fate, and I'll show you a movie about that. And the genome maintenance is the spell check. So growth and survival. So here's an example, a gene called EGFR. It's mutated in several different cancers, primarily lung cancer. There are drugs that now have changed the treatment of lung cancer based on that. And what it does is start that signal process, the ATP phosphorylation of all these proteins down in this chain reaction causing cells to grow. Here are the names of 4 genes that are drivers on the oncogene side and on the tumor suppressor side. We understand these in exquisite detail. So how about cell fate? So here's what cell fate means. All tissues, blood, pancreas, liver, prostate, they begin with a single cell called a stem cell. They turn into progenitor cells, and then they differentiate. So I'm going to show you a movie, and the idea is that there's a set of genes that control this process in a very regulated way. And what we've learned from these sequencing projects is that if one of the genes that controls this goes awry, the cell can't differentiate, you get a problem. So one of the best examples of this is in colon cancer. So this is a movie that my colleague in Holland named Hans Clevers made and let me borrow. And he studies the intestine. So these are the crypts in the intestine. And then what you're going to see is how the stem cell in the intestine, this blue cell, gives rise to progeny, children that move up this escalator to what's called the villus. These are cells that are critical. Their function is primarily to absorb nutrients, food, and so forth. At your homes later tonight, we'll see this process in action. And these cells actually only live for a couple of days. There's massive turnover. And when they get to the top, they've hit the end of their useful life and they die. So what's going to be shown next is what happens if you have a mutation in one of these cell fate genes that's important in this process. And there's a famous gene called APC that does that. And if it's mutated, what happens is now this cell is brown, and it's supposed to keep going up the escalator, but because APC is lost, it can't complete the process, and it piles up, and you get colon cancer. All right. So now that we know this, what can we do about it? How many of these hundreds can we deal with? So lung cancer has been a remarkable success story, and I'll review it really quickly. So for decades, lung cancer has been diagnosed through the microscope. We still do that. Surgeon takes a biopsy. We then look under the microscope, and we see these different patterns, and this is what we call them. With the DNA sequencing, we now know that just that one pattern, adenocarcinoma, has all these different types. There's a portion here we haven't yet figured out, but we know more than half of them. This is way too vague and broad a category. We need to subdivide these into the drivers responsible for them. So each wedge of this piece of pie is how common that mutation is in lung adenocarcinoma. And the good news from this result is that there are drugs either available already or have now been discovered over the last several years that inhibit many of these because they share that property like BCR-ABL of being an enzyme that requires ATP. So in the same way that Gleevec was discovered, inhibitors of many of these have now been discovered. And does it really work? Here's an example of one of the first patients who received one of these pills that inhibits the EGFR. All of this fluffy material, this is a chest x-ray. All of this fluffy material is tumor. And within just a matter of a week, this patient's tumor was clearing away. So this is a remarkable story. We now, when we do clinical trials, we look at the results in these kinds of graphs. This is called a waterfall plot. Each one of these bars is a patient, and what's being measured is how big was the tumor before they started the therapy and how big was it a month or two later. And you can see there are patients who've now had almost 100% shrinkage of their tumor, and a lot of them, but there are others that haven't. And so now a big part of what we're doing in the field is trying to understand if all of these patients have the mutation, how come everyone didn't benefit? And we're getting lots of new insights leading to the combinations of drugs that move this waterfall plot in a favorable direction. What I want to end with is this new form of therapy. I say new even though it was first tried 100 years ago, called immunotherapy. So in order to introduce this to you, I have to remind you how the immune system works. So imagine that you get a viral infection. This is the virus. It gets into the cell. It starts producing viral proteins in the cell. The cell, when a protein is made, it has a certain length of time that it's useful, and then it degrades. And the cell degrades it, chops it up, and it turns out that then these little peptides get presented by a molecule on the surface of the cell. This is called an MHC protein. There's a whole code of the shape of the peptide and the shape of the MHC that's very important in controlling the precision of the immune response. In a similar way, you could imagine that a cancer cell which has a mutant driver, it's not normal. Could the immune system recognize that protein? Does it also get chewed up? Does it get presented on the outside of the cell? We now know that both of these are true. So what do we know about this? Well, a T cell, which is a part of the immune system, has a receptor that very specifically recognizes this interaction. And when that happens, that binding triggers the proliferation of this cell so that it will kill this infected cell. That's how we get rid of an infection. It proliferates, kills the cell. Now, unfortunately, we know because we get cancer that the immune system, if it was an infection, it should have proliferated and killed the cancer cell before it even appeared. But we know that's not the case. So what happened? Well, it turns out that even in the normal situation, there's a built-in sort of fail-safe or backup system to control the immune response. If we didn't have a way to turn this off, the immune system, when overactive, can be lethal. So there are a series of molecules that turn the immune system off once it's been activated. So it goes up and then it goes down. And there are exquisite detail in how this interaction works. Now what happened, and so what we now know is going on, the reason these cancers aren't eliminated quickly is cancer cells have, I don't want to say they're intelligent, but they're devious for sure. They have figured out a way to borrow that strategy and turn off the immune system before it even gets activated. So, what can we do about that? Well, why don't we figure out a way to block this interaction? Can we outsmart the tumor cell? So there are a number of drugs that have recently been approved which go by the name checkpoint inhibitor. One is an antibody against one checkpoint molecule called CTLA-4, and another one is against another checkpoint molecule called PD-1. These two drugs, and there are many versions, different companies have developed them, are the most widely studied cancer drugs today. Essentially every tumor type these drugs are being studied in, and they're now approved for at least 3 or 4 different tumor types. And why? When you give the inhibitor, you disable this part and the tumor cell proliferates and the tumor cell is killed. So the first example of that working was in melanoma. So remember, melanoma is caused by sun exposure. Those are tumors that happen to have a lot of mutations. This is a patient who has melanoma with metastasis in the liver. This patient received CTLA-4 and the melanoma disappeared. One of the most famous examples of this treatment being successful is former President Jimmy Carter, had melanoma with metastasis to the brain. He got one of these drugs, completely cleared. What's really amazing about this also is a subset of these patients are out more than 10 years without a relapse. So the immune system in this case has been taught and is still there. It's a drug that's continually active to pick up any further tumor cells. Now, let's go back to this. So remember, the lung cancers who didn't smoke have a low number of mutations, but the ones that did smoke have a high number of mutations. It turns out that these immune drugs work best in melanoma and in lung cancer, but only in the patients who have a smoking history. So you don't interpret that as it's okay to smoke because there's a drug. The other one, the spell check tumors, the colon cancers, they now also respond to this drug or this category of drugs. So this is late breaking in terms of why this is happening, but the leading hypothesis, and I think it's a very compelling one, is that if you've got a lot of mutations, the probability that you've created a mutation in a peptide that's going to stimulate the immune system better is higher. So I don't want you to, again, having a lot of mutations is clearly bad, but your probability of responding to one of these drugs is increased if you have a lot of mutations. All right, so now we've got surgery, radiation, chemotherapy, targeted therapy, and immunotherapy. So we have an incredibly exciting arsenal. This is why it's so exciting. This is why all of us spend 5 days here talking about the nitty-gritty of how these drugs are working. And I had to extend this because it's just the pace at which these drugs have been developed is startling. So Gleevec was approved in 2001. It wasn't the first targeted therapy, but it was a handful. You could count them on one hand. It was pretty impressive just, 2 years ago. We were up at 35, but I just looked online yesterday, and there are 71 approved targeted therapies. I mean, this is a testament to this massive investment in biology, the sequencing, et cetera. It's also a testament to the power of the biotechnology and pharmaceutical industry to react to this investment in public science and take it to the next level. But, should we, applaud and go home, or what's left to do? So first, I think I've made it, abundantly clear that this has been possible only because we understand the problem in detail that we didn't have, back when the war on cancer was declared by President Nixon. We understand taxonomy. Taxonomy is how we classify tumors. We're using mutations to call patients to determine what cancer they have. We understand a lot more about how resistance is developed, et cetera. But we still have a lot of work ahead. And I want to finish with just a few thoughts on what that work looks like and why I say that. So this is a pie chart that I made together with my colleague David Sollett at Sloan-Kettering. So, and it's based on an investment that at Sloan-Kettering we made about 2 years ago in sequencing patients who have metastatic cancer who we see and treat. So we've done a little over 10,000. And if you look at that, in 2/3 of those patients, we know what caused their cancer. But in 1/3, we still don't. We see a few mutations, but they're not on the driver list. We call that cancer of unknown driver. And we need to figure that out. Otherwise, we're not going to have a good strategy to treat those tumors. And we can talk about how we think we can do it, but it's not trivial. It's a challenge. I think the other problem is that even of the 2/3 where we know exactly what's going on, we have drugs for about half of them. The other half, which we call undruggable, are not because no one cares. I mean, these aren't small diseases where you can't, encourage the pharmaceutical community to get involved. These are super challenging. They're not these ATP-dependent enzymes. We actually don't know chemically how to go after them. But we didn't know how chemically to go after these ATP-dependent enzymes until Gleevec. So I'm optimistic. We just have to keep investing. So my last slide is just to prove that I really am on this panel. We lead quite the glamorous life. This is my colleague, Jose Bazelga. We were both invited to meet with the vice president back in January, but we had to go to Davos, to the World Economic Forum to do it, but I was happy to do it. So, I'll just leave you with the idea that with those last two challenges, there's not a single moonshot project that just finishes the job. It's still complicated. But I'm very grateful to the Vice President for bringing so much attention to the problem. It's really galvanized conversations amongst many branches of government that weren't happening before amongst the pharmaceutical sector and amongst scientists. And I'm an eternal optimist. We are going to continue to fight this. Thank you very much. Yeah.
So Charles has offered to take questions if you have any, and we can use the microphone.
This may be a little off point or even far off point, but last November, my beautiful golden retriever, Buffy, died of cancer. And I believe she had soft tissue sarcoma. Now, I'm wondering if you have in your bag of tricks now something that could have helped her.
I don't know, but I can tell you that soft tissue sarcoma is a human disease as well. There are different subtypes. We do DNA analysis to figure out what subtype, and then for some there are therapies and for some they're not. The dog genome has been sequenced. There's lots of interest in learning from that. So I think another few years from now, probably a different story.
In fact, I was just asked to be on an advisory group who actually want to do exactly for dogs, do a dog cancer genome project for a number of reasons. Well, there aren't the drugs. They have to repurpose drugs for humans, but that can happen and will happen in the veterinary circuit.
I have a question about the cell taxonomy. It would seem that you need to be tested for this genetic mutation to find the correct inhibitor. In doing that, do you find if your mutation is a driver or a non-driver? Because that would determine whether or not there's an appropriate cure. And if so, where do you go to get such testing done?
Well, so there's a couple of questions in there. So a driver versus non-driver, if it's not a driver, we don't think it has anything to do with the cancer, causing the cancer. So that pie chart I showed at the end, that's just the drivers. We have a driver in 2/3 of patients. What we don't have is a drug for each driver, and that we have a drug for half the drivers, roughly. I mean, these are rough estimates. Where do you get tested? So this technology of DNA sequencing is moved so fast that the ability for it to be accessed by the average cancer patient hasn't happened at the scale it should. If there's one moonshot that should happen, it's rolling out this technology much faster to the broader community of cancer patients. It's the top recommendation on this conference call that I am on quite a bit. I don't know if it's going to be adopted, but a specific proposal is that Medicare cover the cost of sequencing for all cancer patients for a period of at least 5 years and collect a database and see what we can learn. You said good luck. It's only $1,000. It's cheaper than a CAT scan.
The people that took the Gleevecs, were they sequenced or were they just given the drug and saw, some people it benefited, some people it didn't, and those people that have benefited, were those tested to see if that was that mutation?
That's a great question, and I'll give you, the answer is that with Gleevec, we didn't have to do DNA sequencing to figure out who had the mutation, because that chromosome test that was done in Philadelphia had become, at that time, a routine part of examining every patient with leukemia. So we knew who had the abnormal chromosome, and we tested the drug only in the patients who had the abnormal chromosome, and we saw a 90% response rate. But the point you make is really important for another reason. That EGFR drug that I mentioned in lung cancer, where the patient's chest x-ray cleared within a week, that happened before anyone knew about the mutations in lung cancer. So the way that story came to light is really good clinicians noticed, well, I've treated 10 patients on this trial and only one of them had that happen. Has anyone seen this happen? Somebody else says, oh, another patient. So 1 out of 10. Normally that would just, kill a drug. You reported that to the CEO of a pharmaceutical company next. But these were such dramatic responses, there had to be an explanation. So after the fact, the patients were sequenced and the EGFR mutation was found, and some of the people who made that discovery are sitting in the back of the room.
Okay.
So I wanted to go back to Buffy, because they just recently did I have a neighboring lab that works on injecting Clostridium into tumors and having the immune system, as he described, take over and start to be alerted that something is wrong in that area of the body. And there was a recent paper that was published that used canines. Their owners identified that they had something wrong with them. They went to the vet, they had sarcomas, and they were injected with Clostridium, and many of them had remission. So that's exciting as well. So there's many ways to attack the tumors using the immune system. So I believe we could save Buffy.
Where does pre-diagnosis sequencing come in? Pre-diagnosis. So to sequence people before they get the cancer in the event that they shouldn't, if they have a familiar...
Sure. All right. So there's two parts to that. The first is, do you have a mutation in your family, in your germline is the genetic term, that predisposes you to cancer? And should we be sequencing everyone to find out if you're predisposed? Well, the answer is not everyone. There's very few genes, sort of somewhat disappointing, in which you can make the case that it's worth doing that. But the most compelling is the BRCA1 and BRCA2. In fact, I'm involved in a prostate cancer project where those genes are much more common than we thought in men who have metastatic prostate cancer but not primary prostate cancer. But there's another way to think about how you can use DNA sequencing and that is, can you detect cancer early, not prevent it, but catch it earlier. So it turns out that as cancer cells are growing, when they die, and the cancer cells do die normally, DNA from the cancer cell is released into the body, and it can circulate in the blood. And these technologies are so powerful that you can actually find the mutations in the blood. Now, whether or not you can detect a tumor when it's tiny and in the blood, that question's going to get, the answer is going to be known in, I imagine, a year or two. It's going to raise another problem if you can find it, because then you do a whole body scan and you don't see anything. What do you do next? But you would then follow the level of this and watch the person more carefully.
Other than just testing the genetic structure of somebody and in the research, are you also looking at the external factors like a smoker, where lead levels change in the body, or you have unbound copper and you have this oxidative stress and adrenal fatigue that all play in to potentially creating the mutations? Does that play into the research as well?
It does, and in fact some people would argue that the best moonshot we could do would be to eliminate carcinogenic behaviors like smoking, sunscreen everywhere, etc. And I tend to agree that we could, if you could eliminate smoking, which is of course an ethical, all these other non-medical aspects to it, the burden of cancer would drop substantially.
So the levels of, like, in a person, when you're looking at cancer, are you also looking literally at the levels of the different metals of the body and how they the synchrony of those metals together?
That's being looked at in a smaller scale and by some groups. It hasn't risen to a sort of moonshot kind of level, and I don't know if the science is, if that's because it's not justified or it just hasn't gotten enough attention.
However, there is people in this room, scientists in this room, who have sequenced whole cancer genomes that can now look at the genome and actually identify, based on the nature of the mutations, what type of what induced those changes. So there's a signature for smoking, for instance, in the DNA, and there's a signature for other chemicals for sunlight and things like that. So those signatures, but it turns out if you sequence the normal cells, you find the same mutations in normal cells, especially in the skin. So the immune system is working in cancer, but one has to boost it up to get rid of it.
Well, the immune system is what I wanted to ask you about. When you showed the different kinds of cancer, that list omitted lymphomas. And since people with lymphoma are often thought to have a compromised immune system, does this sort of targeted approach work on them, or is that why that kind of cancer was omitted from the list?
Yeah, well, it wasn't intentionally omitted from the list. There has been a lymphoma sequencing project. It's interesting you asked, because we heard a fantastic talk about this earlier today. But the answer is that immune therapy can work in certain lymphomas. This PD-1 drug, for reasons that are still somewhat mysterious, has induced complete remissions in patients with Hodgkin's lymphoma who'd failed all other therapies. There's some really fascinating theories as to why. You may know, or some of you will know, that the actual tumor cell in Hodgkin's is a tiny % of the lymphoma, and most of what's happening is the immune system coming in to try to kill that tumor cell, but it's being disabled, and perhaps that's why this checkpoint antibody is working.
Have any of these drugs been used preventatively in patients who have the genetic markers for disease but not yet showing the tumors?
Preventive. There's certain retinoic acids that are therapies that are used as prevention and for head and neck cancer risk in patients who have prior smoking history or alcohol use. There's tamoxifen is a estrogen receptor drug that's used to prevent breast cancer or diminish risk in women who are at high risk from BRCA mutation. Some of my colleagues can maybe help me.
So, I mean, Herceptin, which is a commonly used breast cancer drug, patients have initial treatment and then they remain on that drug to prevent recurrence. That's a common way of trying to extend survivorship in cancer. There are quite a few drugs like that. Gleevec, I think still patients have to take that every day of their life.
There was a report with there was a report just maybe a week or so ago, in a randomized study. You can use one of these blood DNA tests to see how much CML there is in the patient, and if it goes down a millionfold, and then stays down undetectable for 2 years, you can stop the Gleevec, and half the patients will relapse and the other half won't. So, why don't you talk about metformin? No, metformin is a diabetes drug, and patients who've taken metformin for diabetes have a lower risk of getting various cancers, and Dr. Evans knows why.
Any other questions? So I hope you've one up here, Joyce.
This is a very basic question, but there's been some stuff in the news about talcum powder and ovarian cancer. What's your opinion? Is there any connection?
You've stumped the panel. Can I call a friend?
All right. On that note, I think Charles has done a great job in giving you a sense of...
Thank you.
Giving you a sense of that there is a lot of excitement in cancer therapy, but we do have a long way to go. It is going to rely on support for increased science, and increased basic research, but also therapeutic research from the federal government. So if you get a chance, tell your politicians that that is very, very important. But I want to thank Charles. I also forgot to mention that Charles is a trustee of Cold Spring Harbor Laboratory and hence my boss. So I want to thank you for doing this. So we have, for those who are going to dinner parties, to